Welcome to this CyCon machine learning tool tutorial. Today we're going to be talking about the nearest centroid classifier. The nearest centroid classifier assigns class labels to new data points based on the mean or centroid of the closest training sample class. Steps of this algorithm are 1. The centroid for each target class is calculated during training. 2. The distance between a new input data point and the centroid of each class is calculated. And 3. The minimum distance calculated in step 2 is chosen and the new data point is assigned to the associated class. So this is a visual showing what's going on within the nearest centroid classifier. You have these classes with groups of training data points. These centroids for each class have been calculated. Then a new data point is introduced into the space. The distance between this new data point and each of the centroids is calculated, and the shortest distance between this new data point and the nearest centroid is selected, and that data point becomes classified within that class. There are multiple methods for measuring distances within this algorithm. Two of them are the Euclidean distance, which is the straight line distance between points that is denoted by this equation here and this blue line. And the Manhattan distance, which is the distance between points when moving only along horizontal and vertical lines. That is shown here by this equation and this red line. Advantages of this algorithm are that it is simple and easy to use, it uses very minimal computational resources, and it's easily interpretable. Limitations include the fact that it is sensitive to outliers, and it assumes that classes are evenly distributed even when this may not be the case. For our example today, we're going to be using phytomining data. Phytomining being the cultivation of special plant species with the goal of harvesting them to obtain metals from soil. These plant species are called hyperaccumulators, and they can accumulate very high concentrations of minerals without suffering from negative health consequences. So this is a brief look at the example data we're going to be using today. We have data on 2 different species, 7 different varieties within each species, across 5 different blocks, with 2 different tissue types, roots and shoots, dry mass in grams, and the total rare earth element concentration in parts per million within the plant tissue. Now, we need to pre-process the data before we actually plug it into the algorithm, since it is unable to understand textual data within the columns. So for our pre-processing, we have encoded those columns with textual data to have numerical data instead. And for our predicted feature today, we're going to be looking at tissue type. Okay, so this is the CyCon machine learning tool. I have gone ahead and named my project and given a short description of what I'll be doing today with the tool. For my data type, I'm going to select tabular since it's in a table format. I've already uploaded my CSV file with my GRASS data. My target label, like we mentioned before, we're going to predict tissue type. For the method, I've chosen machine learning algorithm, and I've chosen the nearest centroid classifier. For my metrics, I went ahead and chose accuracy. Now I'm going to click perform. So this is my accuracy here. I can also take a look at the y-test values, which are the actual values within the data set, and then the y-predicted values, which is what the algorithm predicted these values would be. I can also take a look at the data in a confusion matrix to see where my data is being misclassified by the algorithm. This has been the CyCon machine learning tool tutorial for the nearest centroid classifier algorithm. Thank you for watching.